Hello, Tuskegee University students. This is Dr. Gerald Griffin, and today I will be discussing major concepts in Week 4 of Biology 315, General Neurobiology. Chapter 6, as we discussed in Week 4, focuses on neurochemistry, particularly neurotransmitters and other chemicals that relate messages from one neuron to the next. Why are we so curious about neurochemistry? Why is there a whole chapter devoted to it in your textbook? Well, it's the actions of those chemicals that allows for all behaviors, all major behaviors, to be performed. In fact, there are numerous books, such as this one written by Professor Larry Young, The Chemistry Between Us, that relates the neurochemistry that allows individuals to feel emotions of love, to be attracted to one another, to feel those quote unquote emotional connections with another individual. Therefore, the neurochemicals that are involved in your nervous system are particularly important not just for basic function, but also higher level functions and feelings associated with humans. What do I mean about neurochemicals? Well, these are just mainly chemicals, molecules that have major actions in the nervous system, whether it be in the peripheral nervous system as well as the central nervous system. We will focus on, for this week, neurotransmitters, which are small molecules, and the major ones that you need to be concerned with are glutamate, GABA, glycine, dopamine, serotonin, histamine, norepinephrine, and epinephrine. The major neuropeptide that you should focus on is substance P, which is highlighted in Chapter 6 in Purves et al. The major purine that is discussed is ATP. That's right, the main molecule you associate with being produced in mitochondria is actually considered a key neurochemical. And then there is the other category that your book talks about briefly. And the one that you really want to focus on is nitric oxide. Well, what do you really need to focus on when you're reading Chapter 6 and zoning in on your studying? Well, for each neurochemical, you should be able to answer fully the following 6 questions. Where is the neurochemical synthesized? Is it being synthesized in the cell body, or is it being synthesized locally in the presynaptic terminal, or perhaps even the dendrite? Number 2, how is the neurochemical synthesized? In other words, what's the precursor? For this course, you don't need to focus on step-by-step reactions, but you should know the major precursor for all neurochemicals that I've just listed. How are the neurochemicals packaged? What type of vesicle are they in? Are they in the small synaptic vesicles, or are they in large, dense core vesicles? What type of stimulation releases the neurochemical? Do you need a long train of action potentials, or is one or two enough for some release of the neurochemical into the synaptic cleft? Once in the synaptic cleft, what types of receptors will the neurochemical bind to at the postsynaptic cell? And lastly, how is the neurochemical removed from the synapse? For example, for many small neurotransmitter molecules, it's simple diffusion, whereas higher, larger neuropeptides, some proteases or enzymes will be needed to break down the neuropeptide. Key neurochemicals, excuse me, key neurochemicals. What you really want to focus on, again, are these neurochemicals that are discussed in detail in Chapter 6. Glutamate. It is probably the most important neurotransmitter in the central nervous system. GABA, which is mainly known for its inhibitory actions, but it's actually been shown that in developing animals and developing neurons, GABA is actually excitatory. You should understand why that is. Also, another major inhibitory neurotransmitter is glycine. One that you should be very readily familiar with is the neurotransmitter acetylcholine. Know its two different types of receptors that it binds to, and whether that's in the peripheral nervous system or the central nervous system. What are the actions on those two different receptor systems? And the biogenic amines. Why are they important, and why are they having in a subclass all by themselves? What are some particular insights that we have learned from studying substance P and other neuropeptides? Purines, especially ATP, is important for actually promoting wakefulness. How does it do that? How does it bind to receptors, and what receptors does it bind to actually have physiological actions in the central nervous system? And lastly, that other category. How is nitric oxide, a gas, even considered a neurotransmitter? And can you actually make the debate that perhaps it's not a neurotransmitter, but simply a second messenger? The goal for week 3 is singular, but I believe it is heavy-handed. You should be able to differentiate the synthesis, the postsynaptic actions, and the removal of key neurochemicals. Understand how they're made, understand how they work, understand how they're removed, and the insights and the effects they have on the postsynaptic neurons as well as the overall organism. Many human diseases are due to imbalances of neurotransmitters. And so I really want to kind of pressure you to somewhat to fully understand these neurochemicals. It's a short chapter, but it's a very important chapter to fully understand how these small molecules have huge effects, not only for cells and the central nervous system by itself, but whole organisms, particularly humans. And with that, I say thank you for your time and have a great day.